Hello everyone, Anna is talking right now. Sure you figured that out by now. We are gonna go and do something a little bit fun. We're gonna go and talk about pathology because who doesn't like to look at gross pictures of things, right? Let's start with a couple of outlines. All right, for a lot of diseases, and you'll explore this more in pathophysiology, for a lot of disease categories, we can kind of split them up into discussing congenital defects. So these are going to be birth defects. And these can be due to different causes. There is the more epigenetic component where something doesn't form right in the embryo as it's being put together. And this is technically often not due to a genetic issue. It's more of an environmental issue, interplay with genetics that causes things not to form right. You can also have mutations that are strictly genetic that cause things not to form correctly, okay? Epigenetic is probably a bigger player in a lot of these congenital birth defects. So basically, environment, something goes wrong in the environment and things don't get put together correctly, okay? The other major category, as I said, is the trauma. I'm also throwing in disease, infection that affects different parts of the spinal cord in here. Now, probably two that you're really familiar with are going to be paraplegia and quadriplegia, okay? So we're going to just kind of put that one there. So para is meaning two, whereas quadriplegia, actually that's an I, plegia, all right, this means four, okay? So this affects the legs, and quadriplegia affects arms and legs, and if it's affecting the arms, it's also affecting the torso to a greater degree. Now, how much of the torso is affected with paraplegia depends on where the injury occurs in the spinal cord. So you've got, with both of these, some kind of trauma that interferes with those tracts in the spinal cord. It could be a crushing injury, it could be a severing injury, it could be a twisting injury, but whatever happens, you are impacting the spinal cord and usually impacting both ascending and descending fiber tracts, although what we probably notice is the loss of motor control. But often sensory indicators are used to figure out the extent of damage. This is more of a pathophysiology thing, so you'll work on that more in that course. Now, some diseases, those are interesting because they can be due to, again, an epigenetic, so we've got over here, we've got disease, and you can have things that are genetic or epigenetic in nature, and that they might have a gene for it, and like for example, a virus triggers the expression of the disease, okay? So some examples of diseases that come on later where that have a genetic or an epigenetic component. Another example of epigenetic would be a lot of our autoimmune disorders. Sorry, I just got distracted there. All right, some examples here would be MS, so multiple sclerosis. And ALS, or Lou Gehrig's disease. ALS stands for amyotrophic lateral sclerosis, which is a big mouthful, so I'm not going to write it out, okay? So multiple sclerosis is interesting in that you're specifically targeting the myelin sheath of that peripheral nervous system, okay? And that obviously is going to prevent this speedy sending of motor and sensory signals, okay? ALS, what's interesting about this one is it's actually killing motor neurons, okay? So multiple sclerosis is affecting the Schwann cells. ALS is affecting the motor neurons, all right? And when you lose the motor neurons, then you end up getting muscular atrophy, and so forth, okay, and you begin to lose the cells, okay. You've also got, so that's genetic, epigenetic examples, but you also have things like polio myelitis, okay, and this is caused by a virus, okay, that gets into the anterior horn of the spinal cord, okay, and it basically causes destruction of the motor neurons there. So remember, anterior horn is motor, okay, so it's destroying those motor neurons, so it's affecting lower motor neurons, okay, and how, so, and then obviously this can lead to paralysis, and sometimes people recover from it and sometimes people don't. How extreme and pervasive the paralysis is depends on where in the anterior horn the virus infected. So was it high up in the neck, in which case it might be affecting your ability to breathe and you end up in one of the iron lungs, or it could be down in the thoracic region of your vertebrae, of your spinal cord, and then it's only your legs that you have to deal with paralysis, okay? All right, now, we did with that whole trauma side. Let's kind of come over here. We're going to draw this out where we've got some more room. And let's look at some congenital disorders. I'll make that bigger so you can see it. Now, we're going to put these with the central nervous system. A lot of these do affect just the spinal cord, but it's hard to say if it's just spinal cord or if it's also the brain. It kind of just depends. The cause, the pathophysiology is the same. The location ends up, or similar. It's the location that ends up changing. So some of my favorites are anencephaly, Spina Bifida, and then you've got meningocele, and you got encephalocele, myelomeningocele. Oh God, do I have to write that? I guess I should if I said it. Meningocele, and then serinomelia. What's interesting about all of these is that they are neural tube defects. So when the neural tube is forming in that embryo, it doesn't close. Okay, so this is what they all have in common. What is different about these is basically location, okay, and severity. So the anencephaly basically is the top, and basically the top of the skull does not fuse in the midline, and then amniotic fluid is going to bathe the top of the cerebrum, and those get eroded away by the fluids. Basically get kind of washed away and atrophy and get destroyed, and so you basically don't have a top of a brain. This one is down in the sacrum area, okay? The meningocele, encephalocele, and myelomeningocele are basically the same thing. It's just where on the meningeal sac. So you're basically looking at the dura mater being affected on these, okay? And then serinomelia is where it fuses the neural tube. It's hard to explain. It fuses midline so that you don't have separated legs, okay? Let me see, what do I have next? Let's kind of look at some spina bifida stuff. All right, next. All right, spina bifida is one of the more common congenital defects, and it has really different degrees of severity. Basically, it's where the spinous process, whether it's a sacrum or a lumbar vertebrae, or it can also be thoracic, spinous process of either a vertebra or sacrum doesn't fuse, all right? So that there's no bone covering that, so you don't have a good vertebral arch. So that basically all you have between the skin and muscle and the spinal cord is the dural sheath, okay? It is actually pretty common, and again, to various degrees of severity. One baby out of every 1,000 or so, okay? Most common locations are usually around L5 to, I don't know, the S3, S4 area, okay, so somewhere in there. This right here is an example of the most severe form. This is Spina Bifida Cystica. And this is basically the dural sheath that's pooking out. It's filled with cerebral spinal fluid. And the reason it's so severe, reason it's such a big concern is because spinal nerves will be in there and those can be compressed. Also, this bag can be ruptured and then all your cerebral spinal fluid leaks out and you die. Okay, so most of these neural tube defects, whether it's Spina Bifida or the other ones, they seem to have a relationship to folic acid deficiencies. All right, so most women know at this point in their lives that if they are planning on getting pregnant or are pregnant, they are recommended to take a folic acid supplement. Also, many things are supplemented or fortified with folic acid, like cereals and breads. And when we went to fortification of our foods with folic acid, and we recommended women planning to get pregnant to take folic acid supplements, the rate of neural tube defects went down, okay? So folic acid, super important, all right? If you vaguely remember from the integumentary system, we talked about how folic acid is damaged by UV radiation. So someone with pale skin in a high UV environment, like New Mexico, is going to have more of their folic acid destroyed than someone with dark skin living in New Mexico. So you pale folks should be taking folic acid. Okay. Let's look at the next picture on Spina Bifida. This one I particularly like because this is your sacrum. All right, and then this is where L5 would be, and here's the articular surface here. Now right here, this should be all solid bone, but what you see is that the bone hasn't formed. So the sac, the dural sheath, can poke out. Now if we come over here and we look at these drawings, you can see here's an example of a baby down probably around S1, S2. And what you have is, this is the mildest version. You have a little indentation where the vertebral arch hasn't formed. Often, for some reason, they have little dimples and little bits of hair that stick out of it. But the tube is still mostly protected. This is the most common Spina Bifida. Now, more extreme, now again, it could be here, 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 here. You can get Spina Bifida anywhere. In this case, they're showing it a little bit higher. And you can see the bones did not fuse at all. And so now the dural sheath can poke out between the bones. And this will be filled with cerebral spinal fluid. This is a moderate severity. This is more severe right here because spinal nerves have gone into the sac, which means if you compress this, you will get paralysis. Both this version and this version are in danger of rupture if it gets smacked or punctured in any way, in which case the pressure of the cerebral spinal fluid around your brain will crash and you've got big problems. All right, a variation of something very similar to Spina Bifida. It's basically the same thing, but we're going to change its name because of where it is and the fact that we're not really dealing with the spinous processes, but it's basically the same thing. So we're dealing with the meninges poking out because the bone did not form in the midline. So this one, encephalocele, meningocele. So the meningocele is going to be between the vertebrae and down here. So, and an encephalocele right here is where you might get brain tissue coming out into that area as well. So they're basically the same thing, but meningocephaly is just dealing with the meninges and cerebrospinal fluid, whereas an encephalocele might involve the brain tissue as well. But you can see on the baby here, it's at the metopic suture. And then down here, it's at the occipital, okay? All right. All right, so mostly I've been showing pretty extreme versions. So like here's one here, and then this one also had an abdominal defect. But you can get milder forms of meningoceles, and I've seen young children with these, and sometimes they end up growing out of them as the bones begin to fuse in the midline as they develop, and then sometimes they have to do surgeries to close these. But here what you see is you've got good bone there, and you've got good bone there. And then right here, the bone is not well formed, and so you just have a very small meningocele that's kind of making a little bit of a dimple that pokes out with a little bit of cerebrospinal fluid. And this is a fairly mild one. All right, so if we get further down, all right, and it's dealing less with the sacrum and lumbar area, we will sometimes change and start using a different name called a myelomeningocele. And this is an example of one of those. All right, this is the scariest one to me. So basically, this is a baby where the top of the skull right here, the bone didn't form, okay? And that exposed it to amniotic fluid. And the amniotic fluid had started to erode the brain tissue, okay? And eventually, all that's left down here is going to be some remnants of the diencephalon and the cerebellum. Obviously, this is not compatible with life. Most children, when they're born with this condition, die immediately, although a couple years ago they had one particular case study that was documented where they kept the baby alive for about a week. This is a picture. So I did a study on a skeletal of a fetus that a neonate who was born in New Mexico and had anencephaly and died, and then their parents donated their body to UNM. Okay, and so I studied this particular baby, and this is the frontal bone. Okay, what's interesting is that the frontal bone should be that shape. So you can see that with the amniotic fluid coming in, it eroded all of that. Up here, this is not a mandible. This is the occipital. Okay, so this is your occipital bone, and it should, obviously, this is where it should be right here. All right, I think I have one more horrific picture to show you. All right, this is an example of cerinomelia. I don't know why there's a red arrow right there. This is also a neural tube defect, but in this case, the defect caused the fusion of the legs in the midline. Okay, so there is no opening for the anus, vagina, or urethra. This is also incompatible with life, and these babies usually die within minutes or a few hours of birth. Okay. All right, take your folic acid. I'm going to go ahead and stop this recording.